Hi there. I'm going to deal with topic 1 in our Science 8 book, and so let's look at living things in the microscope. First of all, it's important to come to a good definition as to what it is that it means to be alive. For example, a little kid might see a bowl of Jello and think to him or herself that maybe the bowl of Jello is alive because when you poke it, the Jello will wiggle and move. So, is it really alive? More importantly, the question is, what characteristics do we as scientists have to help us identify whether something's living or not? Is a log that's fallen over in the forest, is it alive? Or is it dead? So there are 6 characteristics that biologists use to define life. Number 1, all organisms get energy by producing or taking in nutrients. The second characteristic of living things is that all organisms reproduce, grow, and develop. Third, all organisms are made of a cell or groups of cells. Fourth, all organisms respond to stimuli. Fifth, living things have a lifespan and will eventually die, so a life cycle. And the 6th characteristic that all living things have in common is that all organisms produce waste. And so it's important to note these 6 characteristics have to be present for something to be alive. So a bowl of Jello, for example, it'll respond to this one, it'll respond to stimuli, but there are a number of these things that a bowl of Jello does not exhibit, and therefore it isn't alive. Let's talk about a log. A log isn't alive either. It doesn't exhibit a number of these characteristics anymore, but it used to, and so there are 3 categories we can consider. Something that used to be alive, which we call dead. Something that was never alive, and so we call that non-living. And then, of course, the 3rd category is something that is living. And if it's living, if it's a living organism on Earth anyway, it's going to have these 6 characteristics. One of the tools that we need to be familiar with is the microscope to help us study living things in this unit. So here's the scope of how microscopes work. So size really matters here. Our eye can see up to a little less than 1 millimeter of something in size. So a penny, a hand in the human. The light microscope can see somewhere between less than 1 centimeter all the way up to little smaller than a micrometer, which is 1,000th of a millimeter. So to 1,000 micrometers equals a millimeter. 1,000 micrometers equals 1 millimeter. So a human red blood cell is 10 micrometers. A bacteria is 2. And so if you look, a light microscope goes to about there, so it can see bacteria, and that's about it. And electron scanning microscopes can go up to 0.1 of a nanometer. So here's our compound light microscope. We use 2 different lenses to help magnify objects, and they usually range in magnification from 40 to 1,000. And then there's a scanning electron microscope, which, as you can see here, can go from 15 all the way up to 20 200,000 times. Now these are used for viruses and also much better image quality on smaller objects. So let's talk about the parts to a compound light microscope like this one, as well as how we find magnification. We need to know the parts. So the top part is the eyepiece, and it actually has its own objective lens. It usually has a 10 times objective lens. So if we took this off and just looked at something without any of the other parts, it would magnify it 10 times. Then there's the neck, which is how we grab and hold the microscope. Then we have stage controls, and here's the stage, which is where we put the slide. We have an iris diaphragm, which we dial this way or this way to allow more or less light. We always have a light source, and then we have a fine focus knob, which we use at 10, 40, and 100 power, and we have a coarse focus knob. Let's talk about our objective lenses. There's usually 3 on the compound light microscope. There's the smallest one, which is usually 4 times objective. There's a medium one, which is 10 times objective. And then there's the high power, which is usually 40. So let's look at what that really means for magnification. If we use the scanning part with the eyepiece, that really magnifies things 4 times 10, 40 power. If we use the middle one, we take the eyepiece and the middle one, and that magnifies objects 100 times. And if we take the 40 and the 10, that makes 400 for magnification. And let's look quickly at this clip on the microscope.
To begin using a microscope, we first start by cleaning all the lenses with a lens tissue. Then switch on the light. Next, obtain a clean glass slide. Add a drop of water to the slide. Place a small piece of paper on the water. Lower the cover slip gently onto the slide. Place the slide on the stage of the microscope. Turn the objective lens until the low power lens is over the slide. Look through the eyepiece and turn the coarse focusing knob to move the lens away from the slide until the image of the paper is in focus.
Now if you wanted to move in closer than that, you would also use the fine focusing knob to get a little better, finer point of view. So that's a quick overview of characteristics of living things and using the microscope.